Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF Entrance Exam question paper conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. And I have also discussed still previous year's question papers of DBT BET JRF Entrance Exam, and you can find those in my channel playlist. Now next one. Targeted gene disruption cannot be achieved by. Targeted gene depression, it is an experimental replacement of an endogenous gene. Experimental replacement of an endogenous gene with a mutated non-functional version of the gene. So you have got an endogenous gene which is not mutated. You replace that with the mutated form or the non-functional form of the gene. So targeted gene disruption, targeted, cannot be achieved by tDNA integration because it cannot be used for targeted gene disruption. tDNA integration into the plant genome typically occurs through non-homologous recombination, which is a random process. So it cannot be used. Then next one. Centimorgan is defined as the genetic distance between two loci with a statistically corrected recombination frequency of. So if two loci are 1 centimorgan apart, there is a 1% chance that the recombination event will occur between them. So the answer here is option 3, 1%. Then next one. Upon entering the cells, cauliflower mosaic virus accumulates in inclusion bodies in which part of the cell? It is cytoplasm. Answer is option 3. Then next one. Which of the following genes provide herbicide tolerance? It is phosphinothricine acetyltransferase. It has been used, PAT. It has been used to create herbicide tolerant crops. It is used to detoxify the herbicide glufosinate by acetylating it. So this prevents glufosinate from inhibiting the plant enzyme glutamine synthase. And phosphinothricine acetyltransferase, it is isolated from bacteria Streptomyces hygroscopicus and Streptomyces viridochromogene. Then next one. During cell cycle, genome replication occurs in the S phase. Answer is option 4. Plant transformation method that uses tungsten or gold particles coated with DNA accelerated at high velocity, particle gun method. So it is answer is option 2, particle bombardment method or gene gun. Then next one. Which one of the following chemicals enhances Vir gene expression in agrobacterium? It is acetosyringeon. Answer is option 2. In monocot seedlings, the higher concentration of auxin is found in the coleoptile. Answer is option 3. These are just direct questions, that's why I am not explaining much. In plants, which stage of somatic embryo development requires ABA in culture medium? So ABA is usually added to the culture medium during the maturation stage to promote embryo maturation and prepare them for germination. So answer here is option 4, maturing embryo. A classical plant breeder, remember, classical plant breeder wants to develop disease-resistant varieties. What is the first step? First step will naturally be selection of naturally resistant landrace. That is what a classical plant breeder will do as a first step. Natural selection of resistant landrace. Now, which class of enzyme catalyzes the formation of oxaloacetic acid from phosphoenol pyruvic acid in the chloroplast of mesophyll cells. So the enzyme that is involved here is phosphoenol pyruvate carboxylase. So the answer here is carboxylases. Answer is option 2. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion. The major factors influencing the water potential in plants are solute concentration, pressure and gravity. That's correct. Turgor pressure in xylem vessel is responsible for the generation of water potential. Xylem vessels are dead cells. So naturally, water potential in xylem vessel is generated by either transpiration pull or cohesion tension theory. So R is incorrect. So the answer here is option 3. A is correct, R is not correct. Then next one. Which one of the following is the correct sequence of electron transfer in the thylakoid membrane during light cycle of photosynthesis? So pigment system 680 to plastoquinone to cytochrome B6F to plastocyanin to P700. Answer is option 2. Then vinblastine and vincristine, the potent anti-cancer metabolites present in Catranthus roseus, accumulate in which of the following? So they accumulate, this vinblastine and vincristine, they accumulate in the idioblast of Catranthus roseus. Answer is option 4. Then next one. Which one of the following is the immediate effect of ABA dependent stomatal closure in plants? So stomatal closure, it reduces the rate of water loss from the plant through transpiration. So there is reduced transpiration. Then it also reduces the rate of carbon dioxide entry into the plant, which is necessary for photosynthesis. With less carbon dioxide available, rate of photosynthesis also will be less. So reduced transpiration and reduced photosynthesis. That gives your answer as option 4. Then next one. The ABC model of flower development determines organ arrangements in the sequence sepal, petal, stamen, carpel. Due to loss of class A gene function, the observed phenotype will be. So if it is A alone, it codes for sepals. A, B, C classes. A alone, it codes for sepals. A and B, it code for petals. B and C, it code for stamens, and C alone, it codes for carpels. Now A is mutated, so it will be replaced by C itself. So C alone means it will code for carpels. Then C and B, it code for stamens. Again B and C, it codes for stamens, and C alone for carpel. So the sequence will be carpel, stamen, stamen, carpel. Answer is option 4. Which one of the following enzymes is administered to dissolve blood clots during heart attack treatment? It is streptokinase. Answer is option 3. Which one of the following anti-diabetic drug is produced by coupling of GLP-1 peptide with IgG-Fc? It is dulaglutide. Answer is option 3. Then match the enzymes in list 1 with their product items in list 2. Penicillin acylase. Penicillin acylase is an enzyme that catalyzes the hydrolysis of penicillin G to 6-aminopenicillinic acid or 6-APA and phenylacetic acid. This 6-APA or the 6-aminopenicillinic acid, it is intermediate in the production of penicillin, semi-synthetic penicillin such as ampicillin and amoxicillin. So A will be 2. Then alkylase, it is used in the production of bioactive peptides. B1. Thermolysine, it enables the production of aspartame. and it is used in the catalysis. It is used to catalyze the condensation reaction between L-aspartic acid and L-phenylalanine. They both undergo condensation reaction producing aspartame. So thermolysin C4. Beta-galactosidase, it is lactose-free milk. Lactose-free milk is produced by adding beta-galactosidase. Beta-galactosidase enzyme will break down lactose into glucose and galactose. So the resulting milk, it will contain only glucose and galactose, and there won't be much lactose left there. And this will make it suitable for consumption by individuals who are lactose intolerant. So D3. So the answer is option 4, A2B1C4D3. Next question. Given below are two statements. Statement 1. Both aspirin and paracetamol belong to non-narcotic analgesics. So what is non-narcotic analgesic? Are pain relievers that do not contain opioids or other addictive substances. So statement 1 is correct. They belong to non-narcotic analgesics. Statement 2. The synthesis of prostaglandins which stimulate inflammation in the tissue causing pain is inhibited by aspirin. That is also correct. So both statements 1 and 2 are correct. Answer is option 1. Given below are two statements. The manufacturing process of SARS-CoV-2 mRNA vaccine requires synthesis of mRNA from DNA using in vitro transcription. Yes. Now how does this manufacturing process is taking place is the genetic sequence of the SARS-CoV-2 spike protein, it is inserted into a plasmid. The plasmid DNA is used as template for in vitro transcription by RNA polymerase. The resulting mRNA that is produced is purified. The purified mRNA is then formulated with lipids and other excipients, that is sugar or amino acids, to create nanoparticle based vaccines, and then it is administered intramuscularly. So statement 2 we will see the naked mRNA. It is not the naked mRNA that is administered. It is mRNA formulated with lipids and other excipients to create nanoparticle based vaccine is administered intramuscularly. So statement 1 is correct, but statement 2 is incorrect. Answer is option 3. Then next one. The average molecular weight of a nucleotide base in oligonucleotide is 330. So one nucleotide base is 330 molecular weight. What would be the amount required to prepare 100 microliter of 1 micromolar solution of a 20 base pair long single-stranded DNA? So 20 into 330 grams per mole will give you 6600 grams per mole. 1 micromolar is 1 in 10 raised to minus 6 moles per liter. So number of moles needed will be weight by molecular mass. Now leave this number of moles needed. By mistake I wrote it there, but molarity they have given. So molarity is given by the formula 1000 Wb by Mb into V. So 1000 Wb will be Mb into V into M. So upon calculation you will get it as 0.066 microgram. The answer here is option 4, 0.66 microgram. So what I got is 0.066 microgram. If there is any mistake with my calculation, let me know it in the comment section. Then next one. Yeast are facultative anaerobes. They can grow anaerobically during alcohol fermentation and aerobically using oxygen for cellular respiration. Interestingly, wild type yeast cannot live anaerobically using glycerol as they are only fuel source. Which one of the following is the correct explanation? Yeast cannot regenerate NAD plus in the presence of glycerol. That is the correct answer. Why? Because they lack the enzyme to convert glycerol into pyruvate, which is required for NAD plus regeneration through the glycolytic pathway. So 3 is the answer. Now next one. Which one among the following cannot induce cell flocculation? So what is flocculation? It is the process where small particles, cells, bacteria or suspended solids, they will get aggregate and they will cluster together to form larger clusters or flocks. That is called as flocculation. Now which among the following cannot induce flocculation? That it cannot cause aggregation of the cells. Neutralization of anionic charges on the surface of microbial cells. Yes, that will result in cell clustering. Reduction in surface hydrophobicity. Now that will not result in flocculation because hydrophobicity or water repelling property can enhance cell-cell attraction and aggregation resulting in flocculation. Now reducing hydrophobicity means it will make the cell more hydrophilic, so it will tend to interact strongly with water and leading to increased cell-cell repulsion. This would make flocculation less likely to occur. So the answer here is option 2. Use of high molecular weight polymer bridges and alteration of pH also can induce cell flocculation. Next one. The pelleting of microsomal fraction from liver homogenate sample was performed using a centrifuge operated at 15,000 rpm. What is the angular velocity of centrifuge in radians per second? So angular velocity is rpm into 2 pi by 60 for converting it into seconds. Substitute the values, you will get it as 1570. Answer is option 2. Then next one. You have mixed 100 ml of 0.5 molar solution of glucose and 200 ml of 0.5 molar solution of glucose. The resulting solution will contain. Now molarity of both the initial solution is the same, that is 0.5 molar solution of glucose. So the molarity of the resulting solution also will be same, that is 0.5 molar glucose. So the answer here is option 2. The penicillin extraction process involves the following step. Extraction from organic solvent into an aqueous buffer. Extraction from the aqueous buffer into an organic solvent. Extraction of penicillin from filtered broth into organic solvent. Extraction of solvent to obtain penicillin salt. Choose the correct sequence of steps from the options given below. Now coming to answer. First will be extraction of penicillin from the filtered broth into organic solvent. Then comes extraction from organic solvent into an aqueous buffer. Then comes extraction from aqueous buffer into an organic solvent. And then extraction of solvent to obtain penicillin salt. Answer is option 2.